Okay, so I'm going to talk about how to screen cells for the recombinant plasmid. So a recombinant plasmid is just a plasmid that contains your gene of interest. So if we draw the gene of interest right here, let's say, in every one of these cells, right, we have this, that's our gene that we're trying to amplify. A common way to make sure that this gene is present in all of our plasmids that we want to amplify is to use what is called the LAC-Z gene. It's just a partial gene from the lactose operon, which I'll discuss in another video, but it's, if you just want to Google it right now what the lactose operon is, it'll show you what the LAC-Z gene is. So we'll just write it here, LAC-Z. So it's just a small part of the front end of this LAC gene, and the partial LAC-Z gene, it contains a series of unique restriction sites that a piece of DNA can be inserted and cloned. So the bacteria will do the work of the cloning and have the special features that make the presence of the recombinant plasmid evident. So this is a recombinant plasmid. The bacteria that are to be transformed by the plasmid, so as we go through here, it's in a bacterial cell, let's pretend, are LAC-Z minus. So that means that they're missing this LAC-Z gene. So they're missing the front end of the LAC-Z gene, but they contain its back end. Without the plasmid, the bacteria are unable to synthesize what is called beta-galactosidase. So beta-galactosidase is basically a protein or an enzyme that breaks down lactose. So if you drink milk, beta-galactosidase will break down the lactose in the milk into glucose and galactose. So if no foreign DNA has been inserted into the partial LAC-Z gene of the plasmid, the front end of the gene and the back end of the gene will work together with the bacterial cell to produce this beta-galactosidase. But if the foreign DNA is successfully inserted in, then the beta-galactosidase will not be produced. So if we just draw here a LAC-Z minus. So the bacteria that are LAC-Z minus are transformed by the plasmids, and they're plated on a medium that contains ampicillin. So right down here. Only the cells that have been successfully transformed and contain our recombinant plasmid with the ampicillin-resistant gene, so here as well. So this is in pink, this is ampicillin-resistant. So when you put it in an ampicillin medium, obviously it is resistant and it won't die. So only the plasmids that have this ampicillin-resistant will survive on that plate. Some of the cells that contain the intact plasmid, whereas others will contain the recombinant plasmid. So this medium here contains ampicillin, and it also contains a chemical that is called X-gall. So X-gall produces a blue substance when it is cleaved, and the beta-galactosidase will cleave this. So if the beta-galactosidase is present in the bacterial cell, that means that the recombinant plasmid did not get included into it, and it will turn blue. So if we looked at the medium, we would have a bunch of little blue bacterial cells or colonies that are turning blue. But how do we know what ones are our recombinant plasmid that contain our gene of interest? Well, they'll be in white because they didn't cleave anything. They didn't cleave the X-gall, so they didn't turn blue. They didn't have a stained membrane from cleaving the X-gall, and they're also ampicillin resistant, and they contain our gene of interest. So that is how you screen to make sure that your gene of interest is included, and then you would just culture these colonies and grow them on a separate medium in a bunch of different petri dishes, and then you would have a massive amplification of your gene of interest.